Yes, I am live. I think I am live. Dear students, let me know if you can see me, hear me. Please let me know. Yes, can you all hear me? Can you all see me properly? Let me know, please. Yeah, you can keep on commenting. Keep your comments coming in. Okay. Fine, everything fine. Can you all see me? Can you all hear me? Yes, yes, I am live. Is my audio, video clearly visible to you all? Okay, so here we go. Just give a thumbs up if my audio is proper. I can see my video is going fine, but what about audio? Can you all hear me properly and clearly? Okay, here we go. So, welcome to this Sunday, fabulous Sunday morning, and we are going to make this Sunday morning productive also. Today, we are going to draw a few important diagrams which are very important from your examination point of view. These diagrams are from biology textbook, okay, and these are the diagrams. Your biology textbook is full of diagrams. There are so many diagrams, but all the diagrams will not be asked in the examination. There are certain diagrams which you need to practice, which are prescribed for your board examination. We are going to concentrate only on those diagrams, right? So first of all, we are going to draw a duplicated chromosome, and this question was asked in 2014. So most of the question you will see that was asked are PYQs, that is previous year question, right? Okay, so here we have a first diagram which is duplicated chromosome. This diagram is very easy, but it can be asked in the examination. Now about the diagrams, there will be three diagrams. Okay, let us say two or three diagrams. Two diagrams are definitely asked in the examination. Okay, so you have to draw at least two or maximum three diagrams, and this diagram or three diagrams, and each diagram carries 3 marks. That means weightage for your diagram will be equal to 6, minimum 6 and maximum 9 marks. 5 marks is equal to 1%. 5 marks is equal to 1%. So you can see 6 to 9 marks is a huge chunk of your diagram. Hope so that this year we get at least 3 diagrams for drawing. And if they ask us 3 diagrams, we are going to draw all 3 diagrams perfectly, right? So first diagram, remember that certain tips for drawing a diagram. Now I know that most of the students are not good at drawing. Even I'm not that good at drawing, but when it comes to biology or whatever diagram we need to draw for the examination, I can easily and properly draw the diagram. For ICSE board examination, what we require is proper showing all the features. Your diagram must be accurate, neat and accurate. Okay, and beauty of a diagram is not important. Maybe your diagram can look very beautiful, you will decorate your diagram properly, but if the important parts are missing, then you won't be getting marks, right? So please make sure that show all the parts properly. Now coming, okay, this was the exact question, exact wording which was used in 2014. Draw duplicate chromosome and label its part. What is duplicate chromosome? Okay, duplicate chromosome means chromosome with sister chromatids. How do we draw? This is one of the easiest diagram. You can celebrate in the examination hall right then and there if you get this diagram. So here we have a centromere, right? A circle centromere, and this is first sister chromatid, and this one is the second one. Okay, we want duplicated. Okay, so we have to draw two sister chromatids. Okay, and labeling it. This is called a centromere. Now, about the labeling, there are so many myths about labeling. This is centromere. Only remember that where we have to write the names on the all the names should be right hand side, or all the names should be on left hand side. You can keep on writing your doubts, fine. Okay, so whether you write on the right hand side or left hand side, that is not important. But whenever you draw an arrow, please make sure that arrow is towards the part and not towards the name. Your arrow will point towards the part. Okay, and these two are what? Sister chromatids. I hope you can see whatever I have drawn, you can see it clearly, right? So these are the labelings, these are sister chromatids. Is Rajvi here? Okay, now this is what? A chromosome, which is also written as 2n, duplicated. Okay, this part is also called a chromosome. Which part? This part. Okay, but this part, okay, this one and this one is sister chromatid, and this part is what? This is what? Half chromosome. Okay, and this is chromatid. So please, so this part will be very important when we draw phases of karyokinesis. I hope you have understood this. Whenever you draw a diagram in your examination, please make sure that you draw your diagram with the help of pencil and don't use pen for drawing a diagram, right? And labeling should also be done with the pencil. And what you can do is you can draw a box. Okay, box around the diagram. Whenever you draw a diagram in the examination, please don't forget to draw this box. And now you can use your pen to label this diagram. Okay, so what is this diagram called? Duplicated chromosome. Okay, so this is your first diagram with proper labeling, right? If you have any doubt, you can write in the comment box. Please keep on writing in the comment box so I know that you are present. No, that is a chromosome. That is a chromosome, not half chromosome. Even not a daughter chromosome. Okay, this is duplicated chromosome. And all my offline students, okay, they have to show me this diagram when you come for a next class, that is tomorrow. And all the other students, okay, all the other students will draw this diagram and will take a snapshot and send me, right? All my online students. Okay, how big this diagram should be? Now remember that again I will say that beauty is not important. If you draw a beautiful diagram, but if your parts are not properly shown, you will not get marks. Okay, and I will tell you one secret of drawing a diagram, that is, when you draw a big diagram, it is easier to draw because you get a lot of space for showing all the details. So even if on a one full scape you draw one diagram, it is absolutely fine. Let us say that this is your full scape. And if you use the entire page for drawing a diagram, that is absolutely fine. But please make sure that your diagram is what? Proportionate. Length and breadth should be proper. Right? You can ask me your doubts. Fine. So this was one of the easiest doubts. So we can say this to start with. Okay, this was a easy diagram. Let us move on to second wala part. Very important part. Okay, second and very important part. Right, we are going to draw phases of karyokinesis, or these are also called stages of cell division. Okay, stages of cell division of mitosis. Okay, so phases of karyokinesis. Karyo means nucleus and kinesis means what? Division. So title is phases of karyokinesis, or maybe even you can call this as stages of cell division of mitosis. Fine. There are four stages. There are four stages of mitosis. Okay, which you can remember as PMAT. P stands for prophase, M for metaphase, A for anaphase, and T for telophase. Now see how question is asked. Okay, this can be asked in a form of a diagram question also. So diagram will be given, one stage will be given, and you will be asked to draw a previous stage or a next stage, right? Okay, so draw a neat label diagram of First phase of mitosis. First phase of mitosis is prophase. Nowhere they will not ask you question directly that you have to draw prophase. Okay, they will say draw a diagram first. Okay, or draw a diagram which comes before metaphase or first phase of mitosis, having 4 chromosomes. Now see this, every year they keep on changing this number. Okay, here maximum they can ask is 4 number of chromosomes. Okay, 2n is equal to 4. Maybe they will ask you 3 chromosomes also or 2 chromosomes also. So in your board examination, please make sure that you read your question properly, right? And what teacher expects from you. Okay, what teachers expect from you, what are the things which we are going to look out for when we correct your answer booklet. Okay, or your when you we correct your diagram. So again I repeat, beauty of the diagram is not important, accuracy is important. What do I mean by accuracy? First of all, proportion of your diagram, that is length and breadth, should be proper, right? And we will look out for these characteristics features. Okay, all those students who are attending the class right now, don't just attend the class but take out your notebook, pencil, scale, eraser, pen, everything. Okay, as far as possible don't try to use eraser. Maybe if you are drawing this diagram for a first time, you may use a eraser, but eraser should, okay, you should not use eraser or try to not use eraser as far as possible. Okay, so this characteristics features, when I evaluate your paper, I will look out for this parts. Okay, this characteristics feature, if I find this in your diagram, okay, and if your diagram is proportionate and it is properly labeled, I'll give you full marks. Okay, so characteristics features, okay, of prophase is disappearing nuclear membrane. Okay, so nuclear membrane start disappearing. Every cell has a nucleus, and nucleus is bounded by a membrane. So that membrane start reappearing, disappearing. Okay, disappearing. And inside a nucleus, there is one more there is a nucleus of a nucleus inside. Like a cell has a nucleus, nucleus also has its nucleus, which is called nucleolus. So this is also disappearing. Please make sure that, okay, please make sure that um, uh, you write this characteristics feature in your examination. No need to write this characteristic features, but you can write it. Okay, but you can write it now when you are practicing, so that you don't forget to show this part. If number of chromosomes are not given in the examination, okay, if number of chromosomes are not given in the examination, by default draw 4 chromosomes. If number of chromosomes are not given, by default you are going to draw 4 chromosomes, right? Here another one thing is not given, that is what we have to draw, plant cell or animal cell. Okay, what I'm going to do is I'm going to draw both the cell, plant cell as well as animal cell. But if they ask you to draw plant cell, then only draw plant cell. If they ask you to draw animal cell, you will be drawing only animal cell, right? Fine. Even sometimes they ask what is the difference between plant cell and animal cell in this particular stage. Okay, so that also we are going to discuss. Okay, let us start. So plant cell, I'll draw this oval shape. Please be careful how I am drawing this. Okay, oval shape. I believe that if you can draw standing line, sleeping line, slant line, and curve, then my lecture will be helpful for you. You can easily draw a diagram. Okay, so draw this, and please make sure that you draw it properly, proportionally, and we have to label the parts in between. Okay, so this side of your page, left side of your page, you draw this for animal cell, and then comes what? Plant cell. Plant cell has, okay, draw this sleeping line, then curve. Okay, curve at the edge. Let us say this was what? Cell membrane, also cell wall. cell wall and cell membrane. Cell wall. Okay, so first difference between plant cell and animal cell. So plant cell has only cell membrane, cell wall is absent. Here cell wall is present, right? Okay. Then what? Uh, Fine. Now what we will do? We will draw one structure which is present only in the animal cell, which is called centriole. Right now, so see this dot represents centriole, right? And centriole is absent in the plant cell. Please remember this, okay? Centriole and esters. See how I am drawing the ester. First this line, okay, at the center, then left side, right side. Right side, left side. Okay, this esters, again esters are also absent in plant cell. If they ask a question what is the difference, you can write this difference. Okay, and here also centriole and esters, left right, center left right, center left right. Okay, then spindle fibers. Okay, I'll draw spindle fibers like this. Spindle fibers like this, and first draw center one, then go left and right. Spindle fibers will be longer in size as compared to the esters. Here what we are going to have? Here we are going to have just a spindle fiber, no centriole, so no dot. You will show spindle fibers directly appearing like this. We will later on label it. Is it fine? Now what? Nucleus at a center, but what is happening over here? This nuclear membrane, nucleus ka covering, nuclear membrane is disappearing. So draw this line in a membrane in a form of dash, in a form of dash, because membrane is what? Membrane is disappearing. Same manner here also, nuclear membrane disappearing. Nuclear membrane disappearing. As far as possible, avoid using erasers. Even if you draw a single diagram on a single page, it is absolutely fine. The bigger diagram you draw, easier it will be. Okay? And disappearing nucleolus. So for nucleolus, what you will draw? You will draw something like this. Disappearing. Nucleolus is disappearing. Disintegrating. Fine. So these are two characteristic feature: nuclear membrane disappearing and nucleolus also disappearing, right? Okay. And then we have to draw, as we have decided, we are going to draw 4 chromosomes. Okay, chromosomes are inside the nucleus. So this is my first chromosome. Already we have drawn chromosome 1, 2, 3 and 4. Okay, if you have any problem with visibility, you can write in the chat box. 4 chromosomes. When you wrote your pre-boards, I'm sure you must have got this question. Just type in the comment box. So have you got any question related to prophase or metaphase or anaphase and telophase? Okay, write in a comment box which phase you have got, because this is a sure short question, right? Okay, let me label the parts now. First, I will label all uncommon parts. Okay, parts which only animal cell has. One is the ester. Which color should I use? Is this color clearly visible? Ester. Should I write esters or ester? I'll write ester, single. So these backward lines, are esters. Okay. So first write the name and draw arrow. Okay, you got anaphase. Then we have esters only in the animal cell. Then centriole. See, there are so many names starting with C, like centrosome, centriole, centromere, chromatin, chromatid. So this can be very confusing. This part is also present in the animal cell, right? Then common part, that is spindle fibers. Spindle fibers, right? It is present both in plant cell and animal cell. Arrow will be towards the part, right? Then what? Please write, don't write disappearing nuclear membrane only, write complete, okay? Disappearing nuclear membrane. Common in both. Arrow towards the part. Then chromosomes. Okay, chromosomes. Nucleolus. Disappearing nucleolus. Just write nucleolus. If you want, you can mention disappearing. Correct? Please don't forget to draw this box. You can use scale and pencil for drawing this box. This is your animal cell. And this is plant cell. Okay, and title is prophase. Okay, prophase. You can use pen for drawing this title. Okay, you can use pen for drawing this title, but for drawing a diagram as well as labeling the parts, you have to use pencil only. I hope you are finding everything easy, right? Any problem anywhere, let me know. Any problem anywhere, please let me know. Fine. Maybe you have drawn something, but your teacher has given wrong or anything like that. You can type that. Okay, I'll clear your doubts. This is a nice opportunity. Okay. Please make sure that you are drawing this diagram right now. I want to see this diagram. At the end of the class, you are going to send me this diagram. How you are going to send me, I'll let you know. Okay. If they ask you, write the two difference. So first difference is that plant cell has a cell wall. Cell wall is dead, for your kind information. Okay. Then animal cell has a centriole and ester, right? Centriole and ester. So we got 3 points. Plant cell has cell wall and animal cell do not have a cell wall, right? Point number 2 is plant cell. Animal cell has esters. Plant cell do not have that. Then animal cell has centriole which initiates cell division, but plant cell do not have, right? Have we labeled all the parts? Anything anything we forgot or anything like that, let me know. Fine. Can we move on to next question? Can we move on to next question? Yes. Okay. So here we go. Third diagram, which is draw, obviously second part, draw a well labeled diagram to show metaphase. Okay, metaphase in an animal plant cell. As I have discussed, key in 2012 when this question was asked, they have asked only animal cell and not the plant cell. Only animal cell and plant cell. But as we are doing a, this is a practice session, so we are going to draw both. Having 4 chromosomes. Okay, they have clearly mentioned 4 chromosomes. Maximum they are going to ask is 4 chromosome and minimum will be 2 chromosome. I hope I am clear, Hetal. So let us draw both. Okay, fine. So oval shape. Okay, for animal cell, I'll do a better job. You can use your entire page. The bigger diagram you draw, easier it is. And plant cell, first I will draw cell membrane. Then I'll draw cell wall. There should be a little gap between cell membrane and cell wall, right? Okay. Now, what are the characteristic features? Okay, chromosome along the equatorial plane, that is midline of a cell. Okay, now most of the students go wrong here. There is a lot of confusion. You refer to ready-made notes. Okay, where what happens? First of all, you have to understand what is the equatorial plane or what is the midline. Okay, now we are drawing our diagram like a globe, or when we draw earth circular, so like that. So this line is the equatorial plane. This line is the equatorial plane. Now where confusion comes, when you see, when you refer to a diagram, ready-made diagram, or when you refer to any notes, what they do, they draw a cell some like this. So their equatorial plane is this one, right? But we are not drawing a diagram like this. Okay, our diagram is what? Portrait, a kind of portrait, right? Standing, and here it is like sleeping or a landscape diagram. Okay, so don't get confused. बाकी कुछ नहीं करना है. Whatever I have said, just follow that. मैं जो बता रहा उतना ही follow करो. That is more than enough. That is more than enough. Right? Any doubt? Now, as I'm telling you, most of the students go wrong here. Okay? तो क्या करते हैं? पहले हम लोग क्या करेंगे? We will draw exclusive parts which are there in the animal cell. So this centriole and esters. 1, 2, 3, 4. Okay, these are esters, right? Then also here centriole and esters, right? No centriole, no ester in the plant cell. We will start drawing a chromosome. Okay, over here 1, 2, third one, and fourth one along the equatorial plate. Correct? Here also 4. 1, 2, 3, and 4. Now we will draw spindle fibers, right? On these spindle fibers, chromosomes are attached along the equatorial plate. You can draw two extra also spindle fibers. Here no centriole, no esters. Okay, directly spindle fibers. You can use your scale also to get straight lines. You can use your scale. Fine. This is what? Animal cell. This is plant cell. All right, any doubt? Let us start labeling now. Esters, only in animal cell. Centriole, only in animal cell. Now comes the common part, that is spindle fibers. Spindle fibers. Okay, now this is important. Here comes the characteristics features. Chromosomes along, write this, along equatorial. Please don't write completely. Chromosomes along, okay, equatorial plane, right? Anything else? Are we missing out on something? Correct? That's it. We'll draw a box. You can use pencil and scale for drawing this box. And label this diagram, which is metaphase. If you know the diagram, अगर आपको diagram draw करने आता है, so what you can do, इससे related question answer आएंगे, जैसे like they ask you, name the phase, technical terms, name the phase when chromosome align themselves along the equatorial plane. So that phase is metaphase, right? Yes, Yatish, I think you got specimen papers, right, na? Let me know any doubt or else move on to next diagram. Today we are going to draw totally 8 diagrams and make our Sunday a super Sunday, a productive Sunday, right? Can we continue? Can we move on to next diagram? That is fourth one, next phase. They may give you this diagram and they will ask you draw diagram following this one. Okay, let's move on to next wala, that is anaphase. Characteristics feature is sister chromatids separate and are pulled towards the opposite end. Okay, come on, let's start drawing this diagram. As usual, first comes the animal cell, then comes the plant cell, Okay, exclusive parts, centriole, esters. Start with center, then two on the left hand side and two on the right hand side. Then on the opposite side, center, left, right, esters. Okay, no centriole, no ester over there. Okay, you have to draw one more line. Okay, one more border, which is the cell wall. The distance between cell wall and cell membrane will be less. Okay, now what? Sister chromatid separate and pull towards the opposite end. Oreo, do you have any 2021 board question paper? Just write to me. I'll send it. Okay, write in a comment box. I'll send you. Okay, now see 1, 2, 3, 4 spindle fibers. Okay, here also 1, 2, 3, 4. First draw spindle fibers. This will be easier for you. Then draw chromosomes. Pulled towards the opposite end. First draw spindle fibers, then chromosome, sister chromatids. Okay, these are sister chromatids and not chromosomes. Pull towards the opposite end. Okay, the part which I am drawing, that is sister chromatids and not chromosomes. 1, 2, 3, 4. Same way 1, 2, 3, 4. And right? Fine. Now let us start labeling. First, esters, which are exclusively present where? In animal cell. Then centriole, spindle fibers, arrow should be towards the part and not towards the name, right? Spindle fibers are present both in plant cell and animal cell. Sister chromatids, sister chromatids. Arrow will be towards the part, right? Plant cell, use pencil for labeling and not pen. Animal cell, and the title is anaphase. Anaphase. Draw a table. Draw a box around your diagram. Yes, any doubt anywhere? Use scale and pencil so that you can draw straight lines. For drawing diagrams, I am not going to use auto parts. Oh no. Okay. Fine. I hope you are enjoying drawing this diagram. Draw it as big as possible. It will be easier for you. No problem if you draw one diagram in a single page or maximum two. Can we continue? Where it is not correctly labeled. Okay. Okay, I'm so sorry. Okay, this is what? I'm so sorry. This is animal cell. Yes, this is animal cell. And this one is the plant cell. Right. Absolutely right. Yes, thank you very much, Tisia. If I have written incorrect spelling or wrong labeling, please bring it to my notice. Okay? Yes, thanks once again. Can we move on to next one? Now, fifth one is very important. This is the mostly asked question, frequently asked question. I have seen so many school asking this question to draw last phase, which is telophase. Are you ready? Can we move on to next one? Chalo, whoever present, okay, can write, put a thumbs up in the comment box if you are ready for the next phase, that is telophase. Come on, quick. If you have any doubt, you can ask me. Yes, Anya is ready, Mrunal is ready, Deshita, Rajvi ready, Flory ready, Hetal ready. Okay. Fine, let us continue. Let us continue. Let us move on to next one, that is telophase. Right? This is very important. Characteristics feature. Please write this down. This is different from the previous one. We see that in animal cell there is a furrow formation, and in plant cell plate formation. And this is the phase where single cell has two nucleus. There are two nucleus, okay, and these parts are different. These are opposite to the first phase, that is prophase. Okay, here in prophase there was disappearing and here reappearing of nuclear membrane and nucleolus. So these two parts are exactly opposite. You may get a question in your examination related to this, how telophase is opposite to prophase. So in prophase there was disappearing of nuclear membrane and nucleolus, but here both the parts will reappear. Okay, this is not the regular diagram we are going to draw. ये थोड़ा सा अलग होने वाला है, so be careful. Are you ready? Can we start? Yes, so here we go. Draw a diagram like this. For animal cell you will draw a furrow. थोड़ा छोटा बनाता. Okay, like English letter 8. Okay, this is better. Fine. And plant cell. Cell membrane first, and then cell plate. A very small gap between cell wall and cell wall and cell membrane. A very small gap between cell wall and cell membrane. And plate formation. There is a plate. Okay. So this part is called a furrow or a cleavage. Okay, and this is the cell plate, which is forming. Clear? Fine. So this is nucleus ka membrane, which is nuclear membrane, which has reappeared. Reappearing of nuclear membrane. Reappearing of nuclear membrane. Reappearing of nucleus. Okay, so these two parts are exactly opposite of what happens in prophase. So in this sense, prophase and telophase are different. Okay, so nucleus and nuclear membrane, right? So cleavage here, cell plate there. Now no need to draw centriole esters. Okay. Now we have to draw what? Not spindle fibers. They are chromatin fibers. Chromatin fibers. Chromatin fibers. Okay, you are going to love this. Okay, see, pay attention. You are going to love to draw this part. Spindle fibers. Now chromosomes will, okay, will uncoil and you will get, okay, draw something like this. Don't draw too dark. These are chromatin fibers. Okay, sister chromatin separates. Okay, then this is chromatin fibers. Okay, chromatin fibers. Chromosomes uncoil to chromatin fibers, right? First we will draw exclusive parts, that is what? Furrow formation in animal cell and cell plate formation exclusive for plant cell. And arrow should be towards the part and not towards the name, right? Now common to both is nuclear membrane. Nuclear membrane. Arrow towards the part. Nucleolus. Nucleolus is the nucleus of the nucleus. Then chromatin fibers. Okay, these words are chromatid sisters, and this is chromatin fibers. Fibers, thread. Right? Okay, this is animal cell. And this one is the plant cell. In the examination, you may not be asked to draw both, only one of the two. But when we practice, we don't know what will be asked. So we are drawing both. Right? Are we missing out on anything? You can write in the comment box. And title, which is telophase. You can use pen to write your title. I will love to see diagrams drawn by you. So please send me diagram after the class. Okay, how to send, I'll let you know. Right? Now next we are going to do, which is very, very important. Which is very, very important. right? Are you ready for the next one? So we have completed all 4 phases of karyokinesis, or we have completed what? 4 stages of cell division of mitosis. In meiosis, cell division happens in a different way, right? So first one is prophase. They will not directly give you a question, write draw diagram for prophase. Maybe they will say draw a diagram which is earlier or before metaphase, right? Or the first phase of mitosis. Then second one was metaphase. Characteristics feature is chromosome aligned along the equatorial plate. Then third phase is anaphase. Okay, here sister chromatids and chromatin fibers, they are pulled toward the opposite ends. Then telophase, the different one. Okay, totally different. Correct? Any doubt? Now I am asking you a question. Let me see how much prepared you are for the board examination. Next we are going to draw is the structural and the functional unit of DNA. Any idea? Write in the comment box. What is the structural and the functional unit of DNA? Write in the comment box. Chalo, I am waiting for your answer. Let me see how much prepared you are. Mrunal, what no? No doubt or what? Write in the comment box. Structural and functional unit of DNA. Yes, I am waiting for answer. Okay, no doubt. What is the structural and functional unit of DNA? Okay, if you don't know the answer, write I don't know. But let me know, okay? Or am I not audible to all of you? Structural. Gene is the wrong answer. Mrunal, gene is the wrong answer. Come on, Dishita, Rajvi, Dia, Tisya. I hope I have taken your name correctly. Nitrogenous base is not the correct answer. Not the correct answer. Okay. Nucleosome is also not the correct answer. Nucleosome is also not a correct answer. But yes, you are close. Rajvi, you are close to the correct answer. Okay. क्या करोगे एग्जाम में ये क्वेश्चन आएगा तो what you are going to write? All funny answers. Okay, it is nucleotide. Nucleotide. Okay, nucleotide is what? Structural and functional unit of DNA. Structural and functional unit. I am sure you will tell me without taking any time what is a structural and functional unit of excretory system. Everybody knows this. Every Tom, Dick and Harry know this. Write in a comment box structural and functional unit of excretory system. Okay, yes. Dia, nephron. Rajvi, nephron. Okay. Great. Okay. Also remember this. And this is the most ignored answer, but this can be asked in the examination. Please remember that here you have to show basically three important parts, three important components. You can write them down. That is pentose sugar, nitrogenous base, and phosphate group. Fifth wala is not that important. Fifth wala is not that important if you are drawing a nucleotide. Now pay careful attention. Okay, if you don't comment, write your comment now, it's fine. Okay, now pay careful attention. Okay, we are drawing what? Nucleotide. Let us start with phosphate group. Now this is like you in chemistry you study organic chemistry. All right, now in organic chemistry. So base is organic chemistry. Pay attention. First we will draw what? Pentose sugar. Pentose sugar means in a form of a figure with 5 sides. Okay, pentose sugar. थोड़ा बड़ा बनाते, wait, so that we can label it properly. Pentagon. Okay, 5-sided figure is pentagon. Okay, draw pentagon. Now listen carefully. Now this part is very important. Listen carefully. Listen carefully. Okay, now we know about carbon bonding. Okay, oxygen is over here. So write O. Oxygen is over here. And in organic chemistry, okay, later on you will learn this. Generally we write C, but even dot means carbon. We know carbon has 4 valencies. Okay, even you can draw it like this. This dot means carbon. You use this technique when you go in your 11th standard, but okay, तो ये उसी technique से draw करेंगे. Dot means carbon. So this is first carbon, this is the second carbon, this is the third one, this is the fourth one, और ये जगह तो ऑक्सीजन ने लिया, तो fifth carbon is outside. Okay, fifth carbon is here, so write 5 here. Correct? So oxygen here and 5 carbons. This is what? This is pentose sugar. So out of 3 components, we have taken care of first one, which is called pentose sugar. Right? Then how to draw nitrogenous base? So come here. Draw a rectangle like this, which is nitrogen or nitrogenous base. Nitrogen base is also fine. Okay. All right. And we have what? Phosphate group. Okay. And over here, remember this. Ag. Okay, Ag and TC. SMS में हम लोग यूज करते हैं लैंग्वेज टेक केयर. So AG take care. Okay. A stands for adenine. Adenine. And G stands for guanine. T stands for thymine. And C for cytosine. Right? And adenine and thymine, they are bonded by double hydrogen bond. Okay, this bond is hydrogen bond, and A and T double bond. Again, this can be a question, how adenine and thymine are bonded together by a double hydrogen bond, and guanine and cytosine by triple hydrogen bond. Clear? Yes, Geeta Prasad, welcome back. I think you are in 11th standard now. Yes. So how your studies are going on, Geeta Prasad? I think last year, SEM 2, we have drawn a diagram. Diagram for human anatomy. Okay, finals are going on. Okay, will be held soon. Best of luck for your final examination. Okay, Ipshita is also here. I remember Geeta Prasad used to draw a wonderful diagram. Her diagram of heart was fabulous. I hope these students, this current year student will also draw equally good diagram. I will like to see it after the class. Okay, make a box around this diagram and label it. This is what? Nucleotide. You can use pen to write the title. Okay, this is the most ignored diagram. इप्शिता सर मैं कैसे दिखाऊं? Just ऐसे में you have my number ना, so you can WhatsApp me. Right, this is nucleotide. Let me know any doubt. We have covered all three, right? All the requisite parts. That is pentose sugar, nitrogenous base, phosphate group, hydrogen bond. Can we move on to next one? Ipshita, I already have covered a diagram based question related to animal anatomy. You can stay connected. I'll do a diagram based question for this also. Don't worry. Can we move on to next one? Next is again important. I feel ये वाला अगला वाला क्वेश्चन जो है एग्जाम में आने वाला है. Ipshita, I think you are not regular to my channel, YouTube channel. Recently, last lecture was related to location. My last class was related to location. इसके पहले वाला live class जो था, that was related to location. Ipshita, which test you have tomorrow? Can we continue? Okay. Now next diagram, I think you are going to get in your examination. Okay. Which is last paper, I suppose. This is from chapter absorption. Now guess which is the next diagram. It is from absorption. Absorption by roots. What is the diagram? Stoma? No. Which one is the next diagram? Chapter is absorption by root. Yes, next diagram is root hair. Root hair. Okay, characteristics, features. Now root hair, I think this diagram you also had in your 8th standard. You are already drawing this. Yes. So characteristics feature is extension of epidermal cell of root. So epidermal cell is the external, the outermost cell. Okay, extension of epidermal cell of root, and it has a large vacuoles. Okay, large vacuoles. Okay, let us draw this. Maybe you will get this diagram in your examination. Very important. Okay, very important. देखो ध्यान से देखो. It is plant cell. I'll start with cell membrane. I'll start with cell membrane. try to maintain the proportion. Okay, थोड़ा ज़्यादा ही gap रखेंगे because still we have to draw what? This was cell wall, and I'll draw cell membrane. Right? So nucleus, parts are nucleus. Then vacuole, large vacuole. Okay, large vacuole. That's it. And cytoplasm. Correct? एग्जाम में टक टक करके ओके students love to break silence during the examination in the examination hall by drawing this dots, right? Let us label this cell wall, then cell membrane, nucleus, large vacuole, and cytoplasm. And cytoplasm, right? So this is my diagram. Draw a box around it and title, which you can use your pen. Root hair of plant cell. Root hair of plant cell. Okay, have we missed out anything, any labeling or anything? No, I think we have covered this. I know some students, okay, when teacher asks to draw a human brain, okay, human brain draw करने बोलेंगे तो कोई बकरे का brain draw करता है. So no no marks for entertainment purpose. Only for accuracy. Only for accuracy. I don't think so. ये बहुत difficult diagram है. चलो और इसको beautify करते. If you are good at drawing, इसको आप लोग अगर बहुत time है, which you will have. I know biology में बहुत सारा time होता है. Okay, you can draw another one. Okay, you have lot of time in biology. ठीक है? Now comes Last diagram of the day. Any doubt, let me know. Any doubt, let me know. Otherwise, we are continuing. Okay? Just give me a thumbs up in a chat box, comment box, so that I can move on to the next diagram. Okay? Last diagram of the day. Now, all the remaining diagrams will be covered in our next class. Or should I cover it today itself? Okay. Array-tastic. Okay. Array-tastic. Okay, I hope I have taken your name clearly. I see you are regular on my channel. May I know where are you from? I read your comments also. Which school, which state? Okay, let us go on to the next diagram, which is the last diagram of a day. It is again a root hair. It is again a root hair. But this hair, okay, root hair, which would appear when a concentrated solution of fertilizer, it is placed in a fertilizer solution or a strong sugar solution or in a hypotonic solution, okay. Cell which you are going to get is the plasmolized cell, okay. So this diagram, again this diagram is very very important. Read your question carefully. Acha, you are from Maharashtra, VVHS. Acha, who's this then? Okay. I suppose Vasant Vihar High School. Okay. Okay, this is very important. Don't get confused between this diagram. Okay, this is a root hair and this is the root hair when placed in a sugar solution or strong hypertonic solution or in a fertilizer. Last time exam में आया था तो ये fertilizer word use किया था, not the other two words. Okay, last time question was root hair cell as it would appear when a concentrated solution of of fertilizer is added near it. Okay, draw this carefully. Same diagram outline will be same. Plant cell. Okay, we will start with a cell wall. We will start with a cell wall. We will start with a cell wall. I see that बहुत सारे printed books में ये diagram wrong दिया गया. Now when you draw, last time हम लोगों ने ये straight बनाया, but this time ये ऐसा थोड़ा wavy बनाओ. Don't draw straight. Okay, बहुत सारे printed ready-made notes में this diagram is given wrong. Okay, so please be sure whatever you are drawing. Okay. What else? Now see protoplasm. Now what is protoplasm? Cytoplasm plus cell membrane together is called protoplasm. It is placed where? In concentrated solution of fertilizer and plasmolysis. Okay, this is plasmolized cell. So cell wall and cell membrane will condense. Okay, shrink ho jayega, shrunken protoplasm, right? Now here, what is the characteristics feature? Small vacuoles and shrunken protoplasm. Okay, if you don't know what is protoplasm, so cell membrane, हाँ cell wall and this is cell membrane. Last time हम लोग अभी तक कैसे बना रहे थे? Cell wall and cell membrane. There was a little gap, but here cell membrane plus साइटोप्लाज्म जो डॉट डॉट बनाते हैं वो टुगेदर इज कॉल्ड प्रोटोप्लाज्म. Together called protoplasm, right? And small vacuoles. We all know that plant cell has few vacuoles. Animal cell में बहुत सारे होते हैं, okay? Big and few. लास्ट टाइम 3 बनाया था, या 4 ही बनाते हैं. एंड दिस इज व्हाट, न्यूक्लियस. हाँ, ड्रॉ न्यूक्लियस एज अ डॉट. दिस इज न्यूक्लियस. बहुत सारे बुक में न्यूक्लियस वगैरह इधर बनाया है. मे बी दे आर शोइंग श्रंकन प्रोटोप्लाज्म इन दिस पार्ट. ओके, दिस पार्ट. ओके, दिस इज द, बहुत सारे इसमें रॉन्ग मिलेगा आप लोगों को. सो ड्रॉ इट प्रॉपरली. फाइन. चलो, लेट अस लेबल इट. डू वी हैव स्पेस हियर? कहाँ लेबल करते? चलो इधर करेंगे. व्हाट इज दिस? दिस इज सेल wall. Where is cell membrane? ओके, दिस इज सेल मेम्ब्रेन. देखो कितना गैप है. ओके, तो गैप में क्या होगा ये? लेट मी ड्रॉ साइटोप्लाज्म डॉट्स, योर फेवरेट डॉट्स. And here you are going to write hypertonic solution. Strong hypertonic solution. In this case, अगर एग्जाम में फर्टिलाइजर आया तो आई विल राइट fertilizer, hypertonic solution of fertilizer. हाँ, shrunken. Protoplasm. What is protoplasm? Cell membrane plus cytoplasm. Yes, write inside the diagram. Write this inside the diagram. Hypertonic solution you will write inside the diagram क्योंकि वो label ही है. Write inside the diagram. Okay. Box and title. Plasmolized root hair of plant cell. Or just write ठीक है. Okay, that's all for the day. All the remaining doubts, remaining diagrams we are covering in our next class. If you have any doubt, you can ask. You can label this as root hair. Huh? You can label this as root hair. This cell is root hair cell and this is root hair. Difference between this entire is root hair cell. Okay, and this is root hair. Any doubt, let me know. Any doubt, and please remember cytoplasm. Not cytoplasm, protoplasm. Rajvi, Hetal, that day I was not, I could not recall this name. I was talking about this name, protoplasm. Okay. Now tell me what else you want next. What you want in my next class? Okay, definitely I'm going to complete the remaining diagrams, which are again important diagrams. I'll also try to take a live class for diagram based questions. What else do you want? What else do you want? Nothing? So can I end my class here? Okay, just go through all diagrams. Hmm. Now instructions. We started with what? Duplicated chromosome. Then prophase, metaphase, anaphase, telophase, and very important one. अगर मैं पेपर सेट करूंगा तो I'll put this question. न्यूक्लियोटाइड, राइट? अच्छा, ये लेबल नहीं किया. गुड, वी आर रिवाइजिंग दिस. हाइड्रोजन बॉन्ड, ओके, डबल. हाइड्रोजन बॉन्ड, ओके, ट्रिपल. वन, टू, थ्री, फोर, फाइव. Okay, carbon are at this position. Okay, then this is root hair cell. And finally, plasmolized root hair cell. Plasmolized root hair cell. Okay, now students will send me this diagram on my WhatsApp number. और बाकी online student, okay, online student will send on this number. I think you all have this number with you. Okay, online student will send on this number और बाकी के student लोग on my regular number. Right? So can I end my class? Can I end my class? I hope you have done a good job. I enjoyed drawing all these diagram thoroughly. I hope you all have enjoyed this. Right? Chalo, see you all in my next class. Chalo, bye everyone. Chalo, bye.